students. So let us start with our first metabolic pathway, that is a glycolysis. So glycolysis we also refer as a EMP pathway, that is the Embden-Meyerhof-Parhans pathway. This is due to the honor as they are studied this pathway in detail. That's why we refer this as a Embden-Meyerhof-Parhans pathway, or we refer as a EMP. So what is the literal meaning of glycolysis? Glyco, that is a sweet, and lysis means the breakdown or the loosening. Is it clear? This is an amphibolic pathway. Means first five step is the build up process, and second one is the means first five step we require the energy. That's why we refer this as an anabolic pathway, and in next five step energy is liberated, that we refer as a catabolic pathway. And this glycolysis is the best example of amphibolic pathway. Here both mechanism occur, catabolism as well as anabolism. So the pathway glycolysis generally we referred as it is an enzymatic series of reaction in which one molecule of glucose is converted into two molecules of pyruvate with the net gain of 2 ATP. With the net gain of 2 ATP, and that's why here we refer this is an catabolic pathway. It is an energy liberated pathway, net gain of 2 ATP. So glycolysis means glucose is broken down into the one molecule of glucose, that is a 6 carbon molecule of glucose is converted into two molecules of pyruvate with the net gain of ATP. Is it clear? So see here, and once the pyruvate is formed, that pyruvate either undergo the aerobic oxidation or aerobic condition, further metabolism, or the anaerobic pathway. When pyruvate is further metabolized anaerobic condition, that time that pyruvate is converted into lactate. But when it's converted further proceed aerobic condition, then it converted into the acetyl CoA, and acetyl CoA enter the TCA cycle, and we get the then it enter into the electron transport, and the last product of glucose complete catabolism is the CO2 and H2O. When we refer as glucose is completely catabolized to the CO2 and H2O, that time 32 molecules of ATP is formed when it undergo the aerobic oxidation. But when it undergo the anaerobic oxidation, that time either lactate or ethanol form depending upon the cell's requirement. So see here, glycolysis is broadly divided into two steps: preparative step, and second one is the payoff step. So in preparative step, what happened? Glucose first convert into glucose 6-phosphate. Glucose 6-phosphate is converted into fructose. Fructose 6-phosphate is converted into fructose 1,6-bisphosphate, and then fructose 1,6-phosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Is it clear? So this is a preparative step because here energy is required. Energy is utilized step. Is it clear? 6 molecules, 6 carbon molecule glucose is converted into 2 molecules of 3 carbon, that is a glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. That we refer as a preparative step. Why we refer this as preparative step? Preparative step for the energy release. Okay. In next payoff step, what happened? That glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, whatever DHP form, that is a dihydroxyacetone phosphate, that completely convert into glyceraldehyde 3-phosphate by the epimerase enzymes. Okay, so in short, we have two molecules of glyceraldehyde 3-phosphate. One is the glyceraldehyde 3-phosphate, and second is the DHAP, which is converted into glyceraldehyde 3-phosphate. It's converted into 1,3-bisphosphoglycerate, and 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. Here first molecule of ATP is liberated by the substrate level phosphorylation. Substrate level phosphorylation is the phosphorylation process in which phosphoryl donor is the phosphate rich substrate. Is it clear? And then this 3-phosphoglycerate is converted into 2-phosphoglycerate. 2-phosphoglycerate is converted into phosphoenol pyruvate, and then phosphoenol pyruvate is converted into pyruvate. Again here second molecule of ATP is formed by the same reaction, that is a substrate level phosphorylation. Okay, so again as I told you, substrate level phosphorylation means here the phosphorylation is in phosphorylation process. Phosphoryl donor is the substrate phosphate rich substrate, that is the phosphoenol pyruvate. that which is a donor to donate phosphate to the ADP, and we get the ATP. So this is the overview of glycolysis. In next lecture we start the systematic study of each and every step of glycolysis with the formulas. Okay, so here we stop now, and tomorrow we will continue with the preparative step of glycolysis. Okay, bye.